So we are gonna start this session 12. Now all of you know SRM. You understood everything. Everything is super clear. But don't be afraid. In this session, maybe some details that you missed are going to become even more clear. And it's very important that you stop me and ask any question, okay? Because also your questions ask the rest of the help the rest of the class. Okay, so now you know everything. You have your hypothesis, and you know what is SRM. Now what? Now you want to apply it to your project, okay? You want to build your own Srm Essay. One moment. Okay. So we are going to follow a very easy step-by-step workflow of what you would do. Okay? Before we just explain concepts like elements, now we're going to wrap and put everything together. So how to build your Srm Essay step by step. The first thing is to select your protein, okay, which depends on your hypothesis. Okay, say that we were like in that case that we started first thing in the morning of your favorite pathway and you know which proteins you want to monitor. This is the easiest thing. You know what you want to look for. These protein candidates can come from other mass spectrometry shotgun approaches that you did, some biochemical analysis, some things you read in some papers. But then you ask that question that one of you asked me at the very beginning, is that which peptide do I choose? So this is the next step. As we already saw, there are these rules that would be like the classical rules or the logical rules from a chemistry point of view. And this is what people try to get, although it's not always possible. Okay? Always remembering that your peptide has to be unique. Most of the time, you choose these peptides from your own data that you have, because you already performed some discovery experiments, for example, and you have some mass spectrometry data. Here is an example of one protein. Okay, if you go to UniProt and you see download the FASTA file for this protein, is this sequence. In this case, is CDK2. And then you do in silico digestion, and you can see all the peptides that you have that are tryptic peptides that could be your candidates to monitor in SRM. Then you see, okay, out of these peptides, which ones do I see in my mass spectrometer? And the list gets shorter. Okay, this is what we were talking about before. The first thing is that you need a peptide that has a response in mass spectrometry. And then you start to try to apply all those rules we already covered before. And many get rolled out. Why? Because you have a methionine that can be oxidized, tryptophan, you have a peptide with a miscleavage, you have one that is too short, and well, now the long list you had to choose starts getting shorter. You always remember, keep in mind that is unique. And with this, you go ahead. So in general, it's preferred to use your own data. Maybe you don't have, you can go to some public repositories where there is mass spectrometry data. There are MS2 spectras. You can see which peptides have been most frequently observed. And out of these ones, you can see their fragmentation pattern, and you can, okay, this one can fit. So here there are some examples of these repositories that you can find. This is, for example, proteomics DB, proteomics database, in which you can choose one protein, you can see the peptides that were observed, you can click on one of these peptides and look at the MS2 spectra, okay? And you can say, oh, look, here I see a Y13, Y17, and Y20, so most likely these are good transitions to monitor. So this is one way of answering the question of which peptides you want to monitor, your peptide selection. What you would do next is go get those heavy peptides. The same peptides that you chose, you want to get the heavy ones because those are going to be your internal standards. Okay, so just to remind you, heavy peptides behave like their endogenous partners. They behave the same chromatographically. They behave the same in the mass spectrometer, but there is a mass difference. Remember, we were talking about those Y ions will have a different mass, but this is good for us because then we can distinguish them. But the important thing is that they behave the same. So they are our ideal internal standard. So this is just to remind you that the light or endogenous peptide and the heavy peptide coelute. So when we see this, we know that this is our right signal. This is clear for everyone. We talked about this a lot before. And also to remember that this is not a continuous line. We are measuring in dots. We go here, then here, then here, then here. And this is related with the cycle time and with the amount of transitions that we can monitor in one SRM run. Okay, so what we do once we acquire our heavy peptides or we synthesize them, now we have them in the lab and we want to test them. Why do we test them? Well, because we want to see the response in our mass spectrometer. We already had some data from maybe another mass spectrometer from a repository, but now we inject them in the same instrument in which we're going to perform our analysis, and we see what is this response. We can also assess, if this is our goal, the stability of this peptide. We can measure it over time, for example. And we can try to answer the question, is this a good proxy for our protein quantification? Sometimes, for one same protein, we would acquire 4 or 5 heavy peptides, and then when we see how they behave, we end up choosing 2 or 3. Because one, we don't like how it behaves, you know, so it's nice to have those heavy peptides and test them, because then we have the reality of it. So sometimes these chosen peptides don't follow those rules of no miscleavages, no methionines. Sometimes the best peptides are not following those rules, and sometimes one peptide that follows that rule, maybe it's not the best, because the reality doesn't follow sometimes our logic. Okay, what else do we do with these heavy peptides? We make our spectral libraries. Okay, the spectral libraries are the MS2 libraries. So we can inject these heavy peptides in the same instrument where we are going to perform SRM, or in a different instrument. In this case, we take this mixture of heavy peptides. Imagine that we want to monitor 10 proteins with 2 peptides each. So we have bought 20 heavy peptides. We mix them all, because 20 peptides for a mass spectrometer is easy, nothing. We don't need to run 20 times. We mix them all in one vial. We, in this case, inject them in the orbital, and we acquire the MS2 spectra for each one of these peptides. And this is going to be our reference library, what we call the Spectral Library. It is the compilation of these MS2 spectra.
Even if you use another mass spec, you have to use the same fragmentation method. And
yeah, in general, we like to use HCD fragmentation. CID is also pretty similar. And you can also use the same instrument. But in general, the fragmentation pattern will not change that much. Which ions you see and in which rank of intensity should be pretty similar.
So it's not that easy.
And this,
in my library, maybe I don't have a triple top, but
no.
I can run these tests. If
Yeah.
I want to make a SRM in my lab, I have an Oliva.
Yes.
And after I move to a triple top with my library.
We do that all the time.
Okay.
We do that all the time. And that's going to be your reference to compare those intensity ranks. And sometimes it's not going to be the perfect match of the rank, but it's...
But like that you can know if you can perform your assay, at least. Because if you have a list of heavy peptides, but none of them is really good.
Exactly. Then those peptides are not useful.
Okay.
You also need to test the peptides for a very practical reason. That is, that depends on the quality of the peptide that you buy. If you don't want to perform one of those super accurate quantitative assays, but you just want to do some relative quantitation, sometimes you can buy much cheaper these heavy peptides in a lower quality. And you need to check that the peptide is correct and that it gives you a good signal. Sometimes, I don't know, maybe the synthesis didn't quite work well, or it has a lot of impurities, or whatever. So again, always best to use your own mass spectrometry data, but you can check in the public repositories. There is also SRM Atlas, in which is a repository of where you can see SRM essays and SRM measures. So that's also a good source of information for which the peptides that have been monitored of one protein by SRM, they were chosen for a reason. So those are good candidates. Okay, so you chose your peptides, you bought your heavy peptides, you made your libraries, and now is what we properly call develop the SRM assay. Okay, which is, giving all these parameters to the instrument to monitor the peptides that you want to monitor. Okay, so an SRM method is the series of the best SRM transitions for a target peptide with its retention time and the appropriate instrument parameters that we are going to look at now. Okay, this is a MS2 spectra acquired in an Orbitrap, okay, for this specific peptide. And here we have the ions that we see. Here we have a Y15 with charge 2 ion, a Y7. And from here, this is what serves us to decide which transitions we want to monitor. Okay, we test our heavy peptides, we get all the spectral libraries. Now we look at the specific spectra that are in this library, and which transitions would we choose? Where the logic tells us to choose the most intense transitions, because these ones will be easier to monitor in our triple quadruple instrument. Okay, so for example, typically we can monitor 4 transitions per peptide. In this case, I would choose these 4. You can choose more or you can less, maybe not advisable, but very important. Sometimes in the MS2 spectra, the most intense ion is here, for example, a B2 or a Y2 or a Y1. This happens. This is not the case. Even if the most intense fragment is here, you want to discard this region because it has very little information. It's only 2 amino acids or 1 amino acid. That is going to be common to a lot of peptides. So always remember to discard low mass to charge area. Okay, so this afternoon when you are doing this in practice in your computers, you need to have these tricks in mind.
So the notation of the peptide, I need to understand this K point and this point S.
Okay, this is actually this is like a print screen or something. This is how Skyline, that is the software they're going to use this afternoon, looks like. This is a tryptic peptide with a lysine, and after the point is telling you which amino acid you would have in the protein but you don't have. The peptide goes from the S to the K, and this would be the previous, which makes sense because it was used trypsin. And this is the position of the peptide inside the protein. Okay, so you will be doing this. You will be choosing your transitions in the example that we are going to work with in the tutorial this afternoon, using this software from the McCoss lab called Skyline. It's an open source software that you can download and also do it when you go home. Okay, you download it, you install it. We will use this software, or one could use another one, to work with this data. So you are gonna make your own method. You're gonna choose these transitions, and you will also evaluate one result coming from SRM in this same software. This is a print screen of a computer in which Skyline is open, and this is what you're going to see. Okay, here you have your list of proteins. Inside these proteins you have your peptides. Inside each peptide you have the transitions. Okay, you're going to be playing with this. This is just to show you the view. You will see the spectral library associated with these peptides, and here you will be able to say, I want this fragment, and I want this one, and I don't want this one. And then once you have chosen these things, you can export this information into an Excel sheet, and this information is given to the instrument. So then the instrument receives what we were talking about before. How does the quadruple 1 has to be set to filter for these masses here, and the quadruple 3, and this is in pairs. Okay? These are the masses going to be filtered in quadruple 1, in quadruple 2. This is the time that is going to be spent measuring each one of these transitions, the protein name, and the parameters appropriate to break the peptides into fragments. So this is what one would do, send these parameters to the instrument, and then you go to the instrument with your sample and you measure your samples. After you do the acquisition, okay, now we're going to talk about which parameters you need to acquire the data, which is the collision energy, the dwell time, and the cycle time, again. Okay, so the collision energy is the energy that we give to the peptide so it fragments. This is, depends on the method of fragmentation, this depends on the instrument also. In this case, we can tell Skyline which instrument we use, and it gives us the best collision energy for each one of these pairs, because the collision energy also depends on the mass to charge. So not necessarily each peptide will use the same energy. We using this equation that in this case Skyline does it for you. You just tell it the masses and it calculates what is the best collision energy. And this is the parameter that the instrument needs. Then the dwell time is the time that we spend measuring each transition. It's logic that the more time we spend in one transition, the more sensitive we will get. But this is to a point. So a good number, a typical number is 20 milliseconds. Sometimes maybe it's worth it to reduce this number because we want to monitor more transitions. And we know that if we spend less time with each transition, we can put more. Maybe we just want to do a first run for identification to see which peptides we see and which no. Maybe we can put this down to 15, 10. Or the other way around. There is some specific case, for some peptide, we want to put 30 milliseconds. In general, well, sometimes we play with this number, but not too much. Okay, and the cycle time, you know what it is. No? It's the time that we spend measuring all these transitions. Very importantly, cycle time depends on your chromatography, and this you do need to play with, unless you never touch your chromatography and it's very reproducible and you know exactly which is your best cycle time. How many peptides per protein, how many transitions per peptides? Of course, the more the better. If we could, we would monitor each peptide for each protein and all the transitions that are visible. Why not? No? Why not? Well, because we have the limitation of the cycle time. So, you know, these are typical numbers that are used. You know, sometimes some projects, each protein is monitored with one peptide. You know, this is really case to case, like someone was asking me, if you only monitor one protein, why not 10 peptides of this protein? Better. Okay, it's just to remember that the instrument monitors sequentially, therefore we need to calculate how much time are we spending with this. And to remember that we want those 10 points across the chromatographic peak. So, who can tell me then how many transitions can I monitor if my chromatographic peak is 20 seconds? Volunteers? Okay, we have a dwell time of 20 milliseconds. No? How long is going to be my cycle time if I want 10 points in 20 seconds?
Wasn't 100 transitions with the
Yeah. Okay, so yes, so in 20 seconds divided by 10 points, we have those 2 seconds of cycle time. Okay, in those 2 seconds, if we spend 20 milliseconds per transition, we can monitor 100 transitions. And then the number of peptides and proteins, of course, it depends on our project, on what can we do. So this limitation is always thinking in one run, in one method. But then what happens? We want to measure a lot more proteins and a lot more peptides. What do? We can overcome this limitation. So the first obvious thing is do more methods. But what happens that we are using our sample. 2 methods means 2 runs, means double amount of sample. Also problems maybe with reproducibility, time, money. What can we do? We can do schedule SRM. Okay, schedule SRM is because we have an information that we know because we run our sample with our heavy peptides, and we know each heavy peptide in which minute of the chromatography comes out. Of course, we need to be reproducible, because if the peaks are moving from one run to the next, we don't say anything. But if we are reproducible and we give it a certain tolerance, we say measure this peptide in minute 35.3, and then we can say plus minus 3 minutes. If we are very badly reproducible with the chromatography, we can say plus minus 6 minutes or plus minus half a minute. And then instead of measuring that same mass during 60 minutes, is only measure that mass during 10 minutes or 2 minutes. And this is a way in which we can add more transitions in one same method. And because ideally, we want to keep everything in one method, because it simplifies a lot our work. Okay, so we developed the SRM method, and now we go and monitor our samples in our instrument. Everything clear? One thing we need to measure is the response of our peptide in our mass spectrometer. As you heard today and you heard yesterday in the quantification talk, not each peptide has the same response. Every peptide, even coming from the same protein that was in the same amount in the sample, will give higher or lower intensity. So when we do SRM, we need to measure which one is this response and know it. It's something we need to know. Okay, these are two peptides, they come from the same protein, but if you look at the levels of intensity, here in 200 femtomoles we have this intensity, while here with 10 times less we have a lot more intensity, and the linear range goes for a much wider range. So for each peptide we should determine the linearity range. Why? Because we need to measure the peptides within this linearity range. If we want to identify is one thing, but if we want to quantify in this area, okay, this would be our limit of detection. Why? Because we detect our peptide, but we cannot determine the amount, because here the response is flat. The same here where it reaches the plateau. Okay, this would be our limit of detection, and this would be our limit of quantification. We need to make sure that we are working in this area. And this is peptide specific. So for that, we can use the heavy peptides to determine these things. So we can only quantify peptide in its linearity range. We need to determine the limit of detection, limit of quantification. Ideally, this is done with our heavy peptide, but not by itself, in a matrix that is the same, if possible, and if not similar, to our sample. Because this response can change due to the matrix. What do I mean by matrix? If we are measuring one peptide in sample, in plasma, we want to take this, a little bit of this heavy peptide, spike it into a plasma sample, and then measure it. Because the response can change when it's measured by itself in one vial. Okay? And with all these things, we can determine how many femtomoles we want to spike of our heavy peptide in our sample, because will be an amount that we see nicely, that is in our linearity range, etc., etc. We would like that is quite comparable with the endogenous, so we are more or less in the same scale. Okay, we do what we can.
So
it's not always possible.
Can you repeat the last step?
This one?
Yes.
Well, once we have determined the linearity range, we have tested our heavy peptides, if possible, in a same or very similar matrix than our real sample. Then we can determine the exact amount that we want to spike in our sample of heavy peptide. It's just this.
Okay.
The exact femtomoles or the exact micrograms or whatever. We determine this, and then we spike our sample with the mixture of the heavy peptides. Each heavy peptide, not necessarily, most likely, not in the same concentration, because for each one of them, we have determined which one is the best concentration to be spiked.
One question. If you want to measure different transitions from different peptides, from different proteins that are in a very different concentration in your sample, like one in an order of magnitude of femtomole and the other pico, can you still measure the two at the same time?
Yes, because you will spike different amounts of the heavy peptide representing the different proteins.
Okay, so it's like a completely independent essay from one to the other. I mean, it would not be. Okay, we don't have an influence on that.
No, you spike each peptide in your ideal amount that is in the linearity range, etc. But maybe one protein is more abundant than the other.
And if your endogenous peptide is really in a much lower quantity,
It's possible.
Optimal one that
Yeah, it's possible that you are outside the linearity range in your endogenous peptide, and then the quantification is not going to be accurate.
Then you can stop doing separately or not, because this is volatility now. Is it?
Yeah, but if the amount you have is that low, is not much you can do.
Yeah, but if it's bigger.
Because maybe you are just have enough to, if it's bigger, you can dilute. Yes. Okay, so now we have tested our heavy peptides in our instrument. Now we want to test our sample. By now, we have already decided the number of peptides, the number of transitions. We know very well our chromatography. We know which column we are working with, which one is our gradient, the width of our peaks. And we have also measured the concentration versus the SRM response. And now we go ahead. We have our endogenous proteins in our sample. We have already spiked our heavy peptides. And we monitor our samples. So there are now kind of like two things. We want to verify that what we see is what we want to monitor. So identify our proteins, and then we will make sure we are quantifying. For the identification, because we have our reference peptide, we are going to make sure that it's coeluting. We're going to look at the transition rank. This afternoon, when you use Skyline, you will see that you get a parameter next to each transition that is called the dot product. And this is telling you how well it matches the transition rank with your MS2 library. And the peak shape.
Like a score.
It's kind of a score of how similar is the rank of your transitions with the rank that you get in the MS2. You also will look, these things you will be doing yourself in Skyline. You will look at the peak shape, and you will look at if the same transitions are coeluting. The more transitions you have, of course, the less probable is that you have a random match. Okay, so you will see something like this. You can ask Skyline to show you only the endogenous and only the heavy, or you can ask to show you both. Okay, these are different views that I show you now, so when you go this afternoon, you know what you are looking at. You can also tell Skyline to show you the endogenous signals where you have all these transitions in one single line that would be the addition of all these. And the same with the heavy. So sometimes you will see only one signal for the light peptide. This is not because you have one transition, it's the sum of all the transitions. And then the view that you have is this. This is a view that helps you a lot look for coelution. Because you have them separately, it's not so clear. So that's why, we look at this in this way. We have only one signal, but this the addition. And then here, you don't get the name of the transitions, but the name of the peptide. Okay, then we make sure that we have the right peptide, everything is identified correctly, and then how do we quantify? Okay, you will be doing this afternoon also.
This summary that you said, can you repeat again?
Yeah, this is important because sometimes this is equivalent to this. This is a view that helps you see each one of the transitions and helps you compare, for example, the ranking of transitions between the heavy and the light. But once you have seen this, maybe all you want to know is what is the total signal of my peptide. And then what Skyline does is adds up these signals into one.
The intensity.
Yes, into one like this. When you are looking at this is the addition of all of the transitions that were monitored. It's different ways of looking at the same data. Some help you more depending what in that moment you are looking at.
So this is to something the height of the each peak.
Yes.
It's not linear, it's the height.
Yes. Okay, and when we look at this, we are not looking at one individual transition, but at the sum of all of them. It's just another way of looking, but sometimes people get confused with this. That's why I wanted to tell you this before the afternoon, before you are in front of the screen and you don't know what you're looking at. And then we go to quantify. How do we quantify all these measures that we have been doing? One way is exactly this, adding up all these measures. Okay? So this gives us a total intensity or a total area. We can choose what we want to do. And we have a measure for each one of the peptides. In this case, the light and the heavy, the same for each peptide that we have monitored. Sometimes we can decide that even though we measured, in this case, 6 transitions for this peptide, which the more the better, and the more transitions we have is especially helping us for the identification, so we make sure that those are not interferences. But maybe for the quantification, we don't necessarily need 6 transitions. It can happen that we have some transitions. These ones are actually quite nice, but they could be even more noisy. They could be at this level. And we decide that these transitions are only adding noise, and it's going to increase variability more than anything else. And we can say, okay, we're going to remove this. Now we see the right peptide. Okay, now we get rid of this, and we're not going to use this to quantify. So we can add up only the best 3, for example.
But you have to do the light and heavy, or is possible? If you do this, you have to do both of them.
You have, whatever you do to the light, this is a very good question. Whatever you do to the light, you have to do the same to the heavy. The decision is going to be the same because they will be looking the same, but it's possible that this is a heavy peptide and the equivalent light, because the intensity maybe is quite low, then these ones are super noisy. Or even this one. And you decide to keep only one even.
You have this such a difference between transitions?
Yeah, because remember that, do you remember, or if not, I look for the slide that compares this with the MS2. Each one of these signals represents one peak in the MS2. Okay, this is equivalent to this. If this is the Y9, and this is the Y9, and this is the Y7, well, sorry. Well, no, it's okay, Y9, this is the Y11, and this is the Y10. So the transitions, or like each fragment ion, has its own intensity depending on the peptide. Some can, the more signals you have here, the more confident you will be with your identification. But maybe they are not also good to quantify, and that you can decide.
You check first and then you choose this for that.
Exactly.
You have both the information for quality and then to quantify.
Exactly.
You extract what you need.
Definitely.
You can use just one transition for
Yes.
identification?
For identification, obviously, never only one transition, because you know, it's too little to be confident. There are too many coincidences. But once you are sure that that is your thing, you can use the best for quantification. So then we can quantify. How do we quantify? Well, we do the ratio between the light and the heavy. Okay? After we validated the transitions, we discarded the best transitions, we integrate the peaks. Okay? Then we can calculate the ratio light-heavy in each one of our samples. And this ratio is going to give us the quantification. Okay? Once we compare sample A with sample B, each one with its ratio, okay, the ratio of these ratios will give us the relative quantification. Very important. You probably heard in the quantification session. When you work with ratios, you need to work in the log scale. Okay? Remember. Because ratios don't have a normal distribution. It's just here a little note, reminder. Because I put this and it kind of hurts my eyes that I don't see a log here, but that's how I want to tell you. How do we quantify in an absolute way? Well, as long as we know the amount that we spiked of heavy peptide. And for that, then we need to have acquired heavy standards with high purity and high concentration precision. Because as we mentioned before, we can buy different quality types of heavy standards. If we want to know the exact concentration in our sample, then we need to get the very high quality heavy peptides for which the concentration has been measured. And then again, to do the absolute quantification, we need to do a calibration curve with our heavy peptide to know these femtomoles of heavy peptide correspond to this intensity in my instrument with my metrics, etc., etc. And then we can tell, okay, if I have these femtomoles of heavy peptide, I have these femtomoles of my endogenous peptide. Then we can think about, okay, the endogenous peptide amount that I am measuring, that I believe because I have done everything right and because I know how much heavy I put, does it represent the amount of protein that I had in my sample? Well, we have, you know, depending how well we did every step, etc., etc. Okay? Like I said, when we are comparing between samples, it's easy. To give an absolute number is a lot more difficult. Okay, so this is it. Now, if we have absolute quantification, we can go and calculate the copies per cell of our protein in our sample. We have a ratio, light-heavy. We know the femtomoles of heavy that we spiked, and we can, with the Avogadro number, and we can calculate the number of molecules. We know how many cells we started with. We can calculate the number of copies of molecules we have per cell of our protein.
You have to check that the signal of the standard, if the internal standard is correct with your calibration.
Yes,
No?
yes.
But the calibration is not done with the ratio, it's done with the response. The
It's
calibration
previous,
code.
no?
You want me to go back?
Yeah, one before. The calibration is not done with the ratios, but it's done just with the amount.
Yes.
Not,
You
but,
cal-
but you calculate with the ratios then.
You calculate femtomoles or moles or grams, whatever. You calculate amount, not concentration. Okay? The amount. When I have this amount in my sample, I get this response in my mass spectrometer. Then you can go back to that, because you know that this, or this, in this case the heavy, you know that this response corresponds to, I mean, you know the femtomoles you added to your sample.
You do the calibration curve apart from your experiment.
Yes.
You previously do your
Yes.
calibration and then you put your sample with the internal standard and do the quantification using.
Yes.
But you never play with your sample, like you don't test which dilution of your sample is the best for your test.
Yes, you should.
Ah, okay.
You should also.
Okay.
I mean, yes, I mean, for sure you want to know if in your sample you can see your peptides, or it should be diluted or not.
Maybe you saturate your signal.
Yeah. Yeah.
You have to include that in the workflow also, that you have to
Yeah.
check after your heavy peptide.
Usually you have already checked these things.
Ah, well, you have done your discovery.
Yes.
Yes.
But for sure you need to know your sample, the response to your sample, and some peptides maybe you cannot see in your sample and some you do. Maybe you buy 4 heavy peptides, but then in your sample you can only see 2 because are the higher response one. Then you quantify with 2. Okay, so see, oops. Okay, these are just some things to remember, and if you want, you can ask any question. We have time. Yeah.
For the absolute quantification, you use like the number of cells per cell, but it doesn't give you like how much femtomoles do you have.
The first number that you get is the femtomoles that you have. If you know how many cells you started with your experiment, you can go to that, but not necessarily.
But how,
Maybe you don't want to know.
How do you know like how femtomoles do you have? Like, you know the
Because you know how many femtomoles
you put up from the heavy.
you put from the heavy.
can
So
calculate
exactly, exactly.
And the calibration curve, do you run any time or just when you validate the method? When you prove that it's linear and so on?
This depends on the stability of your heavy peptide. If you have a heavy peptide that is very stable, you do it once, you don't need to do it again. But sometimes in the freezer they precipitate. This is peptide dependent. If I was going to repeat an experiment in 1 year, I would run it again to make sure.
Sorry, the correlation between the heavy peptide as compared with the traditional peptide is done by the intensity.
Yes.
Knowing the intensity, you correlate if you
Yes.
have, for example, 3 parts out of 1.
Yes.
No? So you
Yeah.
divide.
It's a ratio.
It's a ratio. Okay.
It's a ratio. And ideally, you want that this ratio, ideally, to be more accurate, the closer the ratio is to 1, the better. Because if you have one signal 100,000 times bigger than the other, the ratios are not going to be as accurate. But you do what you can.
For the calibration, how do you do the
Every run, you make your calibration curve with your matrix, and after you spike your samples, so you run your calibration curve and then your samples.
Yes, but not every run. This is more or less what he was asking. You determine what is your linearity range, what is your limit of detection, and then you run your project. Maybe you are running samples for 2 weeks with different of the one same project with the same calibration curve.
Okay. But the matrix?
If you would want to run more samples in 3 years, depending on the stability of your heavy peptide, this calibration curve can change because maybe you lost sensitivity.
Yes, you lost peptide.
Exactly, and then it's advisable to...
The sample, no? If you lose the heavy peptide, it's degrading, then you overestimate the concentration.
Exactly. So then it's advisable to run it again because you ran it a long time ago and maybe the sample was thawed and freezed so many times. And
the matrix you use to do this is the matrix, the same matrix of the original sample?
Yes.
Or the matrix that is going to go into the chromatography at the end of the operation of the sample?
the matrix that is going to go to the chromatography, you want to test your heavy peptide in the most similar way that you're going to put your samples, the way you're going to measure your samples.
So
if you start with plasma and you deplete the most abundant proteins, you use that to measure your heavy peptides. After digestion, you do the whole sample processing, the same that you would do. Say that you have a lot of samples and you want to measure them all. To test one idea, to test your heavy peptides, you could take 1 microliter of each, no? Because you don't want to use it all because you need it to measure. And this would be your ideal matrix to test your heavy peptides. If you are working with cell lines, it's easier. You just take a little bit and test it.